institutions to provide demanding healthcare courses for global audiences. So on your screen is our founder, CEO, Dr. Akram Ahmed. He himself is a clinical pharmacist and lecturer with over 10 years of teaching and research experiences in Malaysia, India, and Australia, to name a few in various subjects of medicine. His goal is to help qualified healthcare professionals across the globe, pharmacists, medical doctors, dentists, nurses, lab technicians, and physiotherapists, achieve their passions and dreams by educating and training them. So he will be definitely here and taking on your queries. First, we will proceed with the master class that will be done by Dr. Zahra. And now if we talk about achievements on your screens, and we have already proved it in a consecutive way, like recently the CAPS results are announced. So we are managing and we are successfully taking more than 90% success rate. The global reach of Academically is about 50 plus countries. And if we say about how many people have impacted, like 60,000 plus lives have been impacted with this migration process. And the faculties are very qualified if you join the platform. So now let me brief you about today's faculty who's going to take the session is Dr. Zahratul Quresh. Dr. Zahra is a seasoned medicine professional. She has completed her MBBS and MD in pathology from Hyderabad, India. She is currently the head of biochemical genetics at the Sandar Specialty Diagnostic Private Limited. Dr. Zahra has over 7 years of teaching experience in medical field. She is an MOH laboratory specialist and cleared her USMLE steps 1, 2, and 3. Right now, she has associated with Academically as a USMLE and AMC faculty teaching medicine professionals in intricacies of the USMLE exam so that they can establish their medical careers in the US. So I would like to invite Dr. Zahra here.
Hello, everyone. I hope you can see me. Yeah.
Yeah, so
Good evening.
now I'm
Good morning. Good morning. And I hope this is a good learning experience for you all. And yeah, pretty much that was the brief about me. And so should I start sharing my screen or you go ahead
yes,
with
yes,
yours?
yes. I have unshared
Yeah.
my screen. Now you can just take over by sharing your screen.
Okay, let's see. So can you guys see my screen?
Yes, ma'am, it's visible.
Yeah. So good morning, everyone. Thank you for joining us today. I'm Dr. Zahra. So I've been mentoring students for USMLE and various other licensing exams through Academically. And I hope this session also is a good learning experience for you guys. Before we start the session, I would just like to give you a kind of a picture or, a kind of an idea of what you will get as when you join these classes, what you can expect and who should be what you can expect basically, and how you should utilize these classes and these sessions to the best of abilities. So the thing is, like USMLE step 1, yeah, this step 1 exam has basic sciences in it. So basic sciences, this is what we are we are taking sessions on basic sciences. So they are like, multiple subjects, like 8 subjects are there. All first year and second year combined subjects will be there. So the concept of teaching in these sessions is integrated, and concepts will be elaborated. Concepts will be explained that are high yield and that are highly tested in USMLE. Okay, there will be some things that you have to read, and you have to read by yourself, and there will be that also, we will tell you what you have to read, and there will be some concepts that you have to understand in order to answer these questions, right? Please don't mind my writing is not that bad, but here when I'm using the pointer, somehow it comes out something. So just listen to what I'm saying. So the main objective here is to give you the concepts of understanding, so you don't have to memorize most of the stuff. That is one. Number two, we don't take separate physiology, biochemistry, pharmacology, and all the subjects that are tested in basic sciences. We don't go about all separate subjects. So everything is integrated and combined. So the relevant physiology in that topic, the relevant biochemistry in that topic, the relevant pharmacology will come, and then followed by the pathology. So we complete all the topics, all based on all the subjects that are there on that. So it starts right from anatomy to pathology, and then if it is like behavioral sciences also, we do the same thing. And then there will be some topics separate, like ethics and biostatistics, and all those things will be there. But basically the chunk of the matter will be all these concepts integrated. So you will be like in one topic, you will be rotating among all these. So it will not be separated, because in the exam they are asked together. Like in one question they might be asking the anatomy of the thing, or they might be asking a kind of a drug question. This is not a one reasoning exam, this is a second level reasoning exam. So what is going on, you have to already know. Nobody will ask you how it is going on, and where and why. These are the things that you have to learn for this exam. So basically this is what the learning process will be at academically, because it is an integrated concept wise. And when you take this exam anywhere, a student, an MBBS student who has passed second semester, about going into the clinical rotations around the third year, from that point onward, anywhere in your career you can take this exam, step 1. Okay, the reason I'm saying step after the second MBBS is because in the first MBBS you have this anat, physio, biochem, and the second MBBS you have all that pharma, micropatho, everything. So basically the basics you have to finish, and only then you can attempt this exam. After that you can attempt I think you can attempt many times. But the thing about this is, if you are planning a career in US, or if you're planning a good, I mean, if you're planning to get the best out of this exam, then I would say don't take attempts. Like you should not be in the situation to repeat it. Do it once, do it good. So prepare yourself once before giving the exam. Step 1 exam is pass or fail, pass or fail. So there is no score here, but the score matters in step 2, because all the matter that you read, understand, learn in step 1 will be retested in step 2, and then some more. Okay, and then some. So then the score comes. So how you do and how your subject matter is in first, although it is passed, you might be passed, but if your concept is not clear, your score of this will reflect in step 2. So it's better that you prepare step 1 from the beginning. Okay, so with that said, let me start with a brief introduction of this class. Somebody said something. I can't see any other window here when I when I'm not seeing the Google screen. Okay, so if you guys are leaving any messages, I'll not be able to check that during the session, maybe after the session. And please mute your mics. Please mute your mics. Bhati, you can mute everyone. You can do that. Okay, so this is pretty much the thing. So this is a short topic. It's highly tested and it's high yield. I selected this topic just to give you an understanding of the type of teaching that happens. So if you are comfortable or not, and how you understand these kind of things. So to proceed with this, we have a cell membrane, right? We have a cell membrane. It is a phospholipid bilayer. It's a bilayer cell membrane. In that bilayer cell membrane, that is basically made up of carbohydrates and lipids, and basically lipids, and then there are some carbohydrate moieties, and then there are some protein moieties also in the cell membrane, right? So we are discussing about the protein moieties in the cell membrane, which are which span across the cell membrane, and these make up the transport channels. So there are channels in the protein in the cell membrane, and these are made up of basically protein. They are basically made up of protein, and these channels carry molecules across the cell membrane, So they act as carrier proteins, and then they carry different forms of ion, carbs, and different forms of protein. They act as receptors, they bind to ligands and sell signals into the cells. So they act as enzymes and they bind to substrates. They are selectively permeable. They open and close based on voltage or in response to chemicals. So these are some characteristics of these channels. So there are two types of, basically there are two types of transport systems. One is the active transport system. This is the active transport system, and one is the passive transport system. The passive transport system is the one that doesn't need energy. You can just remember that. And active is it needs energy in the form of ATP, energy in the form of ATP. So ATP is the currency. ATP is the energy currency, and all of the active transports, they need ATP. So ATP is very important. And passive transport, they don't need energy. So they can just, use the gradient. They usually use the gradient of high to low, high to low. Wherever, there is a gradient, then there is a transfer. So passive transport is also known as diffusion, and it can be simple and it can be facilitated. Simple is somebody said something. I'm sorry, I'm not able to hear this. So there is simple diffusion that usually can happen through aquaporins directly through the membrane, or it can use a channel protein. So that is simple diffusion. Then there is facilitated diffusion that uses carrier proteins. Carrier proteins are present in the membrane. They carry and then they help in diffusion. So these are the two types of diffusion. So diffusion basically happens from high to low. There is one more process called osmosis. There is one more process, it is osmosis. That usually happens across a semi-permeable membrane when there are high number of solutes on one side of the membrane, and there is the osmolality of this solution is high because of high number of solute. So this is high, right? So water flows from low to high. So this is the concept of osmosis. It will flow from low to high. So this happens in osmosis, then to dilute the solutes that are there in the membrane. But diffusion happens from high to low. This is also happening in the lungs, where we have alveoli. Alveoli have oxygen from outside, and then there is blood. That oxygen needs to go inside the blood, right? So this process is also called as diffusion, because the high concentration of oxygen in the alveoli, low concentration in the blood, and so this is happening. This diffusion is happening there. So you need to know where how this diffusion is happening in the body, where all it is happening, and the basic mechanism of diffusion is this. Okay, so this is passive transport. Then you will be given this kind of graphs in the exam. They will give you there will be some molecules that are transferring across the membrane, and they will give you this graph, and then they will just label this, and then they will say what kind of diffusion is happening here. So if you see simple diffusion like aquaporins, they use they work on simple diffusion. So they just go along as the concentration of the substance increases, the rate of diffusion also increases. But in facilitated diffusion, because it is using a channel, because it is using a carrier protein, right? So this molecule has to go through this protein, this carrier protein. So the molecules, what they do is when they use this, they tend to saturate the carrier protein, and then when they saturate the carrier protein, it takes time for it to diffuse into the cell. So that is the principle behind facilitated diffusion. That is why it as the concentration increases, first it happens, it keeps on happening, it goes forward, and then when the saturation of the carrier protein takes place, then it plateaus down. So that shows that these carrier proteins are saturated with the ions or whatever it is transporting. So this is the facilitated difference between facilitated and simple transport. This is the difference. And and any molecule that is using carrier proteins will follow facilitated diffusion. Okay. Now the difference of is if it is using energy and it requires a transport protein, then it is active transport. If it is not using energy and it requires a transport protein, it is facilitated. If it doesn't require transport protein, the thing. Now, facilitated diffusion, as we said, it can be voltage gated. So voltage gated works on the mechanism that there is a gating mechanism that opens and closes, and so the ions will go along the gradient. So this is again diffusion. This is the mechanism of how a carrier protein will carry protein, and then it binds to the channel, and then it goes out through the channel. So GLUT receptor work on facilitated diffusion. So whatever you learn here, you have to apply this when you are answering the questions. Facilitated diffusion and active diffusion. So basically they will give you specific molecules and what they will how they will transport, and then they will give you the electrolytes, or they will give you glucose, and then they will ask you what type of diffusion, what type of diffusion is happening here, or active or passive is happening. This is an example of ligand gated channel in which a ligand or a chemical ligand is actually guarding the channel, and then that is used to open the ligand gated channels. The chemical is used to open the ligand gated channels, and then the ion enters, and then it creates a membrane potential, and then that's how muscle contraction happens. This is an example of acetylcholine membrane channel, how it works on the receptors. So again, this is also facilitated diffusion. Now, coming to active transport. Active transport can be of two types. The primary one is the one that uses ATP, and the secondary one is if some pump is already using ATP, it augments the energy there. So a nearby pump can, a nearby channel can use the energy from the primary and then transport the ions. So basically, this is the difference between active and secondary active. So primary active will actually be the one that is using the ATP. Secondary is using a pump that is using, or an already an energy derived process that is being happening. So that's how a secondary transport takes place. So transport of this type occurs in the intestinal epithelium, epithelium of renal tubes, all exocrine glands, gallbladder, and membrane of the. So this is very important. So the concept of these classes is these. After the class, the PPT which is used to take this class will be uploaded in our learning system, where you can refer to the material that has been discussed, and of course the recording also will be there, so that if somebody missed the class, they can see the recording later on. That will be there, and this reading material will also be there. You can read later on, make your own notes, and whatever books you are referring for step 1, this will always be an adjunct to that, and then you can accumulate all your notes there. Okay, so this is the basic example of primary active transport, sodium potassium ATP base pump. This is very highly tested in USMLE. So it uses ATP for the transfer of for the exchange of potassium and sodium. So sodium goes out and potassium comes in. So you can remember this with a mnemonic: pumpkin. So pumpkin is a mnemonic. So K is in pump, K in. So always potassium goes in. So that's how you remember. So if you are getting confused what goes in and what goes out, because this pump is very important, although there are many other pumps but not highly tested, this pump is very important. So you need to know what goes in. So potassium goes in, pumpkin. This is one mnemonic to remember that. It operates on ATP basically. And the application of this pump, I will come to the application. First, I will just go with the secondary active transport. So in secondary active transport, like there is sodium and glucose. So there is energy given by another pump that is there using that energy. So this is the energy or the gradient by one created by the concept of the secondary one. So it can be co-transport together going inside, or counter transport, one going in, one going out. So that is the mechanism of secondary transport. Now, the importance of this pump, sodium potassium ATPase pump, is in cardiac cell. So there is a co-transport happening of there is a co-transport happening of calcium. Co-transport of calcium is happening along with this pump. When sodium goes out, calcium goes in, and increases the calcium troponin, and that helps in contraction or increase in inotropy of cardiac muscle. So basically, this pump usually works, and the pump usually is blocked by a drug called digitalis. It is blocked by a drug called digitalis. So when you block this pump, and so there is you augment the calcium inside, the calcium doesn't go out, you increase intracellular calcium, that leads to increase in cardiac contraction, increase in inotropy. So indirectly, digitalis is working for increasing the inotropy by increasing the calcium, but actually doing what? By blocking this pump. So this is the application of this pump, which works on active transport. So when the question is asked about this, it can be anything. It can be what type of transport it works on, what is the co-transport ion with this, what is the drug that blocks it, and how this. So method is called high yield. So this is how you have to read anything and everything around that. Should be integrated at the same time. Okay, so this is how. Now coming to signal transduction. So what is signal transduction? So the signal transduction is basically, we have a cell the cell has DNA, right? The cell has DNA in it, and all the information is there on the DNA. So whatever function needs to happen in the body has to come out from the instruction has to come out from the DNA. The instruction has to come out from the DNA. But in order to give what function is required, so there has to be a signal that comes inside and tells the DNA, please do this, we need function. For example, after a meal you need insulin, so you have to give a signal to this cell or the DNA that we need insulin because we have taken food, we need all this glucose is there and we need insulin. So that then the DNA gives the information that, okay, this is how you make insulin, and now come on, insulin, go out of the cell. So all the dogma of signal transduction is this: first you get a signal to the DNA, and then from there the work happens. So this is what we are going to discuss, how the signaling in the cell works through gene expression. So it goes to the DNA, and then the gene replication expresses through replication, and then through transcription it goes and forms RNA, and from that translation into proteins, and all this process works. So this is what signal transduction is. Now, the types of molecules that are used in this signal transduction can be electrolytes and ions. They are using ligand gated channels. Okay, basically. And then there are some um, lipid soluble molecules, and then there are some water soluble molecules following different types of signal transduction pathways. Okay, basically these are the major ones which you need to remember. So lipid soluble molecules, the receptor is basically lipid soluble. How they work? The basic concept is, okay, this is the nucleus, this is the cell, this is the nucleus, and this is the DNA in the cell. And the molecule, the hormone or whatever it is, comes and binds to the receptor intracellularly. It changes conformation, and then it goes and gives signal to the DNA to forward the function. So this is the concept of lipid soluble molecules. And all the lipid soluble, like steroids, calcitriol, thyroxines, retinoic acid, they work through this concept. They work through this. Then there is a water soluble concept. Water soluble molecules water soluble concept. And in this there are receptors in the cell membrane. Okay, there are receptors in the cell membrane. This is the cell membrane. So or inside the cell also, sometimes their receptors are there in the inside the cell, and sometimes they are in the cell membrane, or sometimes they are like, in going through the cell membrane, and then the receptor is actually inside the cell membrane, outer side. So cell membrane, and also a part of it is inside the cell membrane, so it's like a combination. So these kind of things will be there. We will discuss how and what are these. So, and then they work on the concept of second messengers. They use second messengers, these water soluble molecules. They use these second messengers, activate protein kinases. So what are protein kinases? Kinases are something that will attach a phosphate, that will give off a phosphate. And phosphates are, high energy molecules. Like in ATP, if you see ATP, we have three triphosphate, right? And ADP, we have diphosphate. And AMP, we have energy that molecule is carrying. Kinases, they activate by giving phosphates. So these are the protein kinases. They are activated, and then that is giving the signal. They control the gene expression through second messengers like cyclic AMP and all these things. We will discuss that. And the examples of these are insulin, glucagon, catecholamines. Basically, they use the water soluble pathway. Okay. Now coming to the mechanism of water soluble proteins, how they use, there are two types in this. Now we are discussing about the water soluble ones, the insulin and all those things, how they are using. So one concept is they use G protein on the membrane. Okay, there is a protein called G protein on the membrane used, and that gives a signal to the second messenger that activates the protein kinase, whatever is the protein kinase, A or C. So basically our purpose is to activate the protein kinase who is going to give the signal to the DNA. Okay, in these kind of signal transductions, protein kinase is the one who's going to do that. So these proteins, they use second messengers. Okay, and what are the who are using this? Glucagon, catecholamines, cholinergic vasopressin, histamine, oxytocin, all these things. You have to know this. You have to know which molecule is using which pathway. First, how the pathway is working, and then which molecule is using which pathway. So they'll give you a question in a clinical vignette, in a scenario in which catecholamines are given to a person, like epinephrine is given to a person, and so the action of epinephrine, whatever clinical signs and symptoms the patient is having, so this is using which pathway. That will be the question. So they want to ask you the biochemical give around these kind of inbuilt protein kinase activity. They have inbuilt protein. Now here, if you see, we are activating a protein, and the other kind, these receptors, they are already having an inbuilt kinase activity, protein kinase activity. So this is the second type. In this second type, we have cell membrane tyrosine kinase. So this is the kinase. See, this is the kinase. What is the protein kinase used here? Tyrosine kinase. These are insulin receptor used by insulin receptors used by insulin. Then we have cytoplasmic tyrosine kinase. So this is inside. So if you see, it will be like this. It will be the membrane will the receptor will be on the membrane, and then inside the cell there will be a tyrosine kinase. So that is cytoplasmic tyrosine kinase, or non-cell membrane, non-membrane or cytoplasmic tyrosine kinase. These are JAK-STAT, used by the growth hormone, JAK-STAT receptors used by the growth hormone, and insulin receptor used by insulin. So this is membrane tyrosine kinase. This is non-membrane, also known as cytoplasmic. Okay, so this is the concept. Now coming to the G protein concept, what we have, what is the concept of the G protein? So G protein has alpha, beta, and gamma. These three subunits will be there in a G protein. G protein, which is inactive, which is when it is not used. Then what happens is it becomes activated, and then after when it becomes activated, the alpha subunit is used by adenyl cyclase, and then that is the one that is actually giving the function, the alpha subunit. The beta and gamma are just keeping it stabilized. Again, after the function, they combine, all of them combine, and then they become inactive. So this is basically the function of GTP. It's a 7-helix membrane spanning the domains. So the material that is written here will be for your reading later on. Okay, so protein kinase, this is what I said. Protein kinase is the one that is actually activating and deactivating. So there will be like when it is it will provide the phosphate for phosphorylating the enzymes and proteins, and then the phosphatase will be removing the phosphate and making it inactive. Okay, so but there are some enzymes that are working in the dephosphorylated state also. That is also there. Some exceptions are there. You don't have to go through those exceptions now, but basically the mechanism is phosphorylation and dephosphorylation. Phosphorylation is done by kinases, dephosphorylation is done by phosphatases. So taste, if you think something that is breaking or removing, phosphatase removing phosphate. This is very easy to remember. So this is how the genes and the enzymes are regulated by shifting into these two forms. Now, pathways that are using the G protein. So there are 3 G protein pathways. GS is the stimulating pathway, GI is the inhibiting pathway, and GQ is another novel pathway. So this is also a type of thing that you have to know the function of that. GS and GI basically are similar, stimulating and inhibiting. So this is they act on cyclic AMP. They act by increasing cyclic AMP or decreasing cyclic AMP. Okay, so you can just remember very easily, increase or decrease of cyclic AMP. Who is the second messenger here? Cyclic AMP is the second messenger in this pathway. And so this second messenger, the protein kinase uses A. Protein kinase A is used here. Here also same. So GS and GQ are basically the same pathways. One is stimulating and one is inhibiting. Okay, the same thing. And the receptors that are used, that are using these pathways are beta 1, beta 2, beta 3, dopamine, histamine, and vasopressin. These are the receptors that are using this pathway. And the receptors that are using the inhibiting pathway are alpha and muscarinic receptors and dopamine D2 receptors. These are the receptors. Now GQ pathway we will discuss now. This is the GQ pathway. So this is also known as the IP3DAG second messenger pathway. Okay, IP3DAG second messenger pathway. So this again is, if you see, this is a type of G protein, right? Alpha, beta, gamma. GDP with alpha, beta, gamma. This is a G protein. G protein gets activated, only the alpha part is used, the gamma beta is not used. This alpha part is actually activating phospholipid PIP2. Okay, so PIP2 will break down this phospholipase. Phospholipase C is a molecule which breaks down, PIP2 breaks down into DAG and IP3. Okay, into DAG and IP3, these two molecules. And this IP3 goes into the endoplasmic reticulum and signals the calcium, which is the second messenger here. Calcium is the second messenger in this pathway. Calcium comes out and then activates the protein kinase C. The protein kinase in this pathway is protein kinase C. Okay, so this is the function. This is how the this is a bit just go through the process. So if you just go through the chain of the process that is happening, then it is it's not that complicated. It looks like it has multiple molecules going on there. So IP3, this question is asked in USMLE. Who activates the calcium? The second messenger, IP3. Always remember, from this to IP3 is the one that is working through calcium. DAG is not working through calcium. DAG has its own power to activate protein kinase by itself. Okay, DAG has its own power to, this is the protein kinase, the green one, this is the protein kinase molecule, protein kinase, this is the protein kinase. So DAG has some power to activate the protein kinase by itself, but the other product IP3 uses calcium to activate protein kinase, but this is more stronger. Okay, when it uses calcium, it is more stronger. This is not that strong. So we have PIP2, which is going into forming DAG, and you don't have to remember the all the diacyl glycerol, blah 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 blah, phosphatidylinosytol, la, 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 no need. Okay, just remember the short forms. Just remember the, there are some words, big words in biochemistry, like HGPRT enzyme, right? You don't have to remember the whole form of this. It is used as HGPRT only. In the exam also they will refer to as HGPRT, and if they are referring to some very big name starting with H, it will invariably be HGPRT. So no need to remember the big names. So these are the things. And IP3, so IP3 is very important. This is the one that activates calcium. So they go into the detail, nowadays they want to make the exam very hard. They go into the detail, what is activating calcium, what is not activating calcium, who is activating protein kinase, and then further what is happening. So this is the mechanism, and this is all the reading material that comes with the matter. You can explain that. Now coming to lipid soluble, so that was about water soluble. So water soluble, that is the only thing that is applied to water soluble hormones. So all steroid hormones, all androgens, estrogens, glucocorticoid, cortisols, or steroid hormones, all these things, they work on this concept. So what is this concept? The molecules go inside the cell, because they are lipid soluble, right? And the cell membrane is a phospholipid bilayer. They can easily, the lipids can easily go through the cell membrane. The only challenge is the water soluble. The water soluble cannot go because it is a lipid membrane. That is why they need channels, and then they need proteins. So the lipid solubles, they go through inside, and then there are some receptors inside, sterol receptors, steroid receptors. They are inside, they are also sleeping, they are also switched to it, so that they don't keep doing their function without any reason. So they have to be like sleeping mode. So they have some shock proteins or something that are keeping them in, like, sleeping mode. When the molecule of interest comes, the shock proteins and all the, the molecules that are inhibiting these receptors, they come off, and then they form a ligand with these molecules, and then they go into the DNA, and then they give the signal to the DNA, please go and do your process. So basically this is the whole end process which we need. We need function from the DNA. We need the function from the DNA for the protein, or whatever function we need in the body right now, that has to come from the DNA. So this is the lipid soluble hormone process. Now coming to non-receptor tyrosine kinase. So we said that in water soluble also we have two, right? In water soluble we have one was the G protein, and one was the inbuilt protein kinase. Inbuilt protein kinase, we have the receptor tyrosine kinase and the non-receptor tyrosine kinase, right? So the non-receptor is this one. The non-receptor tyrosine kinase is the JAK-STAT pathway. In that there is this receptor, and if you see the JAK molecules are spanning inside the cytoplasm. So they will go phosphorylate. Again, phosphorylation is the important process here. Phosphorylation will make them active, and then after getting activated, the JAK and the STAT, they get activated basically, and then they go and give the instructions to the DNA for transcription and all that. Growth hormone works on this pathway. So this is the pathway that there are different subtypes. Janus kinase, this is the name of the thing. There are different subtypes, and this is how this is the explanation of this. Okay, polycythemia vera is a condition that mutation, that leads to mutation of this JAK2 mutation that happens in polycythemia. Controls growth of cells happens, and then so it leads to myeloproliferative polycythemia and myeloproliferative disorders. So basically this is the polycythemia and growth factors. Basically growth factors, polycythemia, erythropoietin is like a growth factor, or EGFR, and all those growth factors, they use these these receptors normally, but when it is mutated, then it gives rise to uncontrolled growth. So this is the pathogenesis of polycythemia vera. Okay. Now, receptor tyrosine kinase. This is the last one. The receptor tyrosine kinase, that was the non-receptor. Receptor tyrosine kinase. In this, the receptor that is inside the intracellular part of the receptor. There is an extracellular part of the receptor, and then there is an intracellular part of the receptor. Whenever the signaling molecule like insulin, this is insulin coming and attaches to the receptor, it goes and gives a signal to the inside part of the receptor, which already has tyrosines, and then they get phosphorylated, they get autophosphorylated, and then this goes and gives the cellular responses that we require. So this is basically the mechanism of receptor tyrosine kinase pathway. They get autophosphorylated, and then this is how insulin is working. And basically this is the procedure of that. So receptor tyrosine kinase also has two pathways. Okay, there are two pathways of receptor tyrosine kinase. One is the RAS MAP kinase pathway, and one is the IRS PI3K AKT insulin signaling pathway. Okay, again, you can just remember AKT. It doesn't even have a full form starting from A or K or Z. This is just protein kinase. This was a protein kinase B something that was named AKT for some reason. I don't know, for God knows what reasons it was named AKT. It doesn't even have a name starting with A or K or T in it. Nothing like that. So it is a AKT insulin signaling pathway. Tyrosine kinase receptor pathways are two. We'll just go quickly towards this. So insulin receptor substance will be there. It activates that. And PI3K, again, this is the one that is using PIP2 to PIP3, and PDK1, which is activating AKT. So this is doing the functions, different functions of glucose of insulin that are coming. There is one more function of PI3. The one more function of PI3, as soon as insulin comes and attaches to these receptors, these are the series of reactions that are going on. I have put it in a very simple form, although there are many other molecules which are irrelevant for this exam, which are not required for you. If you go and Google this receptor, you will get like so many other subunits of these molecules, and all. Don't get confused in that. So we will give you the information which you have to read and which you don't need to read. So this is as much that is required. So PI3K is also responsible for giving the signal of GLUT receptors, which are there in the cytoplasm, to go to go to the periphery to so that glucose can go inside the cell. And basically, GLUT4 receptors for skeletal muscle and adipose tissue. So one of the functions of PI3 is this. And through AKT, it does all these glycogen synthesis, glucose utilization, glucose production, glucose uptake, all these things of insulin reaction happens through this molecule. So it's a bit complicated, but basically phosphorylation of tyrosine kinase is happening over here. This is the tyrosine kinase phosphorylation, and after the phosphorylation, these are the chain of events that is happening from the insulin receptor. The other receptor tyrosine kinase is the RAS pathway. Okay, so in this also there are phosphates in receptor tyrosine kinase. Phosphates are there on this receptor. Okay, these phosphates are basically receptors. Some growth factors, they use this receptor, and then they phosphorylation of this will activate RAS. RAS is a protein. They activate RAS GTP, and then RAF. Through RAF and MAP kinase, this again growth factors, or wherever growth of cell is required, growth factors are used. So this is normally happening, and this is normally happening during the cell cycle when RAS is actually giving the cell. In the cell cycle, we have like G1 phase, and then we have S phase, and we have G2 phase. These are the phases. Then M phase. Phase is the checkpoint phase, where all the proteins are checked, and checked so that they don't go, I mean, unchecked into the S phase, because anything that goes into the S phase will be replicated. Mutated cells are there, then it will be replicated in the S phase. This is, you know, S phase has no points to check there. So that will be incorporated into the DNA. Since the replication of DNA happens in the synthesis phase, so whatever goes through the G phase, it will be replicated, and then the whole cycle will release these kind of cells with these replicated DNA, mutated DNA or normal DNA. So here the G1 phase is regulated by P53 and retinoblastoma, right? RBG. These two are the factors regulating the G1. And cyclins are regulating, cyclins and cyclin D and all are regulating the G1 phase. Multiple things that we have another class for that. We have a separate class for that. But just to briefly give you, the cyclins are the one which are depending on activity of the RAS. The RAS activates the cyclins or deactivates the cyclins that will go into the growth phase or the replication phase and all that. So mutation of RAS can be seen in some kind of, carcinomas, hepatic and all those carcinomas. BRAF is seen in melanomas and all these kind of carcinomas and all. So this pathway will be tested for you in like RAS oncogenes. RAS oncogenes are activated, and this is how this pathway works. And this is for like oncogenes and cancers and all those type of questions. This pathway will be tested, but not in usual like, with insulin. It is never tested with insulin. This is totally different function from, and that insulin is a totally different function, but they both work on the same kind of concept, receptor tyrosine kinase pathways. Okay, so pretty much that is it from my side. I hope that was a good learning experience. Over to you, Bharti. I have to stop sharing my screen. Bharti?
So I think there is some questions from Dr. Vinesh Kumar. Hi,
Okay.
Dr. Zara. Dr. Vinesh, can you please unmute yourself? Dr. Vinesh Kumar, can you please unmute?
Please answer your questions. Give your questions. I'll be happy to answer your questions if anything is there.
There is one student asking, ma'am, please explain USMLE pathway about exams. Ashwarya, she's asking in the chat box.
Okay. Yeah. I can see that. So, pathway about exams. So there are 3 exams: USMLE, Step 1, Step 2, and Step 3. The Step 1 exam you can take anytime, after uh, first or second MBBS. Second MBBS, you can take the Step 1 exam.
And then second, USMLE 2, when they can write, or USMLE 3?
So USMLE 2 usually people take after final year, because the subjects are final year subjects like OBGYN, medicine, surgery, pediatrics, basically all that final year subjects are there. So
So we
ideally
can
taken
say
after
when
final.
they are doing their internship, the right time to write this USMLE part 2.
Yes. Right time. That is the right time. When you are doing your internship, you can take the Step 2 exam.
And USMLE Step 3?
Step 3 is required when you are starting your residency, when you already are into residency, first year of your residency, you can do that, or before applying also, you can take the Step 3 exam anytime. Anytime after you are ECFMG certified, you can take that, but the maximum time is after joining resident. They will only tell you that after joining residency, within first year you should take it. And if somebody has taken before that, they will be a more suitable candidate. Like, they prefer programs, and programs prefer candidates who have already passed Step 3 residency.
Dr. Zara, in the US, so is there any fee to do this PG? They need to pay for any fee to the college?
No, this is like this is a residency program, like a
Yeah.
PG program. So
Yeah.
when you enter, like for example, you are choosing medicine, internal medicine, you match into internal medicine, the program pays you a stipend.
Okay. So it
So
is
first
free,
year,
and even a student will get the
yeah, it is
stipend.
free. is free, you will get a stipend. First year stipend will be some $63,000 based on the college. Some give more than that also. Second year it will be little higher, like 65,000. And third year, if you when you are a senior PG, you will get a little more. So it is like that. Grade wise it is there. So just like a system in India also, when you are doing PG, you are getting stipend, right? Some government colleges, it's like that. So this is a residency program, and when you join any residency program, they will give you stipend. You don't have to pay anything. You just have to pay for the exams only.
Okay.
Okay, yeah. She has raised her hand, but not. Have you muted everyone so that they are not able to unmute, or what? No, no. If we have
Hello?
centrally muted, yes,
Yeah, this was centrally muted. Good morning, ma'am.
yes. Morning.
I wanted to ask that there's a direct pathway, like there are four pathways that when you Google the pathways to go to Australia and study there, since my brother's currently studying in Monash University, he's pursuing a dual degree. For medical field, it is different. There's one with the PLAB, the standard pathway. There is
That is
a
for UK.
I
uh,
mean,
for
Lab is
Australia.
for UK. For Australia,
what
AMC.
are you? I mean, you are asking. I mean, this is a for Australian Medical Council in general, or a specialist pathway. If someone has maybe 10-year experience after MD or MS in India, and there's another pathway called competent pathways for US graduates and UK graduates, not for you.
Okay. That is for the
For you,
yeah,
only the
completely.
option, not the that extended pathway. And through that, you need to pass AMC Part 1 and Part 2. Or someone completed MD or MS from India, and they have very rich experience, like 8-10 years experience, then they can apply directly for the specialist pathway.
And for the direct registration with the Australian Medical Council, we have to give the Australian Medical Council exam.
Yes.
And how long does this take? Like like if I apply this year, all the steps happen within 1 year, or like it takes
This
like
is
It is,
USMLE.
it is,
This
it
is
is
not
up to
the
you.
AMC exam.
Anushka, she's asking regarding the AMC. So
Yeah, but the
it is up to you. I mean, you pass part one. Within 3 months, you can write part two. So if you pass both together, within 6 months, then obviously you can migrate in within 1 year. But if you are not able to pass, then it may take more time. So it's up to you.
okay,
that is a separate course, but AMC, not
Hmm.
the USMLE.
okay. Thank you.
Thank you.
We have classes for AMC as well. So
Okay,
AMC for AMC one exam. For that, we have classes and sessions for that separately.
okay, ma'am. Thank you.
Okay, I think there's no question. Then we can finish here. Thank you so much, Dr. Zara.
Thank you, everyone.
Guys, कोई question है तो पूछ लो baby. I mean, otherwise we are winding up.
Okay, thank you.
Thank you.
Thank
Yes,
you, everyone.
AMC, yesterday itself we did one webinar, so we always did. Next week